Okay, that leads us to cooperative enzymes, which are enzymes with multiple subunits and multiple active sites. And they are in either a low affinity tense state, which is called T, or a high affinity relaxed state, which is R. The graph for the kinetics of cooperative enzymes are S-shaped, like the one right here. So cooperative enzymes have this feature that when more substrate bind to the subunit, that basically encourages the other subunits to make a transition from T state, which is the tense state, to the R state, which is a higher affinity relaxed state. So you can think about it like this. Okay, imagine that the enzyme kinetics of cooperative enzymes like a party, and as more people start arriving at the party, the atmosphere becomes more relaxed, and the party seems more appealing from people outside of the party. But when people start leaving, more people follow to leave because the party becomes less appealing and more tense, if you will. And last point that I want to go over for this section is the numerical value called the Hales coefficient, which basically measures the cooperativeness. So if the value of Hales coefficient is more than 1, that means positively binding is occurring and less than 1 value is negatively cooperative binding. And if the Hales coefficient is 1, that means independent binding. So it's very simple. 2.4 is a short section that talks about effects of local conditions on enzyme activity. Enzyme activity means enzyme velocity or enzyme rate. So yeah, they're all synonymous. So enzyme catalyzed reactions tend to double in velocity for every 10 degrees Celsius increase in temperature until the ideal temperature is reached. For examples, the ideal temperature for the human body is 37 degrees Celsius or 98.6 degrees Fahrenheit. So anything above that temperature will cause the enzyme to denature, and so the activity of the enzyme becomes zero very quickly. But also some enzymes do have this ability to basically regain their function when they are cooled down. pH is also important for enzymes, first because it affects the ionization of the active site, and it can also denature the enzyme. The optimal pH for a human body is 7.4, and anything less than that is called acidemia, which is a life-threatening condition. But again, we do have exceptions in different areas of the body. For example, pepsine, which works in the stomach, has maximal activity around pH of 2. And then we have the pancreatic enzymes, which work in small intestine, and at maximal they are maximal active around a pH of 8.5. Last thing mentioned in this section is that if the levels of salt increases, it can disrupt hydrogen and ionic bonds, which will cause conformational changes in the enzyme, and it can even sometimes denature the enzyme. Okay, and we are done with section 4, and here are the study questions. Section 5 is about regulation of enzyme activity. So we have feedback regulation, which is when enzymes are subject to a regulation by a product in the metabolic pathway. And we also have feed forward regulation, which is when the enzyme is regulated by a metabolite that was made previously. So basically, the metabolite that was made earlier in the pathway regulates the enzyme. So in the case of feedback regulation, we have feedback activation and feedback inhibition. And feedback inhibition is far more common. Uh, Feedback inhibition is also known as a negative feedback, which helps to maintain homeostasis. As you can see in this high concentration of product D inhibits enzyme 1, which causes the whole pathway to slow down. So product D can do that by binding to the active site of enzyme 1, which would make the enzyme unavailable for use for other substrates. Um, this is in fact how our hormonal feedback loops work in our body. Then we have reversible inhibition, which are four types, which are competitive, non-competitive, mixed, and uncompetitive. So let's start with competitive inhibition. So in competitive inhibition, an inhibitor binds to the active site of the enzyme and basically occupies the enzyme and doesn't let the substrate bind to it. Um, I think it's called the competitive inhibitor because if the substrate concentration is higher than the inhibitor concentration, it is more likely for the enzyme to bind to the substrate. So I think about it like a competition between the enzyme and the substrate, and whichever has the higher concentration will win the race, if you will, and bind to the enzyme. Um, it is important to know that the competitive inhibitors do not change the Vmax value, but they do increase the Km value since more enzymes are needed to be able to reach half of the maximum velocity. And here is a Lineweaver-Burke plot for competitive inhibition. And as you can see, the Vmax is the same for both lines, which is the intersection of the y-axis, and the x-interception is the Km value, which has been increased. And just remember that the x-axis on the left represents -1 over Km. So it's like the opposite of the regular graph. So if you go to the left more, you're actually getting a lower value. Next, we have non-competitive inhibition. I guess it's called that because inhibitors and substrate are not competing for the active site. And that's because non-competitive inhibitors bind to the allosteric site instead, which changes the conformation of the enzyme. So the allosteric site are non-catalytic regions of the enzymes that bind regulators. So in this case, unlike no matter how much substrate you add, it will make a difference because they're not even competing for the active site. And by the way, they can also bind to the enzyme substrate complex. So if an enzyme is already bind to a substrate, they can still the inhibitor can still bind to the allosteric site and change the conformation. Since adding non-competitive inhibitors completely takes over the enzyme, therefore it would be less enzymes available, which makes the Vmax to decrease, but it does not change the Km value because the enzymes that are still available will still have the same affinity for their substrate. And again, here is the Lineweaver-Burke plot of enzymes with and without non-competitive inhibitor. As you can see, same Km, but different Vmax. Okay, so mixed inhibitors, they can bind to either enzyme or enzyme substrate complex, but they have different affinity for each. Because if they had the same affinity for both of the enzyme and the enzyme substrate complex, they would just be like the non-competitive inhibitors because they also bind to the allosteric side. So both of them bind to the allosteric side, but the mixed inhibitors have different affinity. So mixed inhibition does have an impact on the Km value, but the affinity of the inhibitor determines the if the Km will go up or down. So basically, if the inhibitor has more affinity for the enzyme alone, so it means it likes binding to the enzyme more than binding to the enzyme substrate complex, that would increase the Km value. But if the inhibitor prefers to bind to the enzyme substrate complex, that will decrease the Km value. But either way, Vmax is always decreased. And lastly, we have uncompetitive inhibition, and they only bind to the enzyme substrate complex. And what they do is that they lock the substrate inside the enzyme and don't let the substrate leave the enzyme. And they do that by binding to the allosteric site of the enzyme as well. Uncompetitive inhibition decreases both Vmax and Km value. And because of that, the Lineweaver-Burke plot would have parallel lines, as you can see here. And this is a table that was provided in the book that it basically compares the reversible inhibitors that we just went over. Okay, so just remember that the inhibition methods that we just talked about were all reversible. But we also have irreversible inhibition, and the difference is that they will make the active site unavailable for a long period of time, or they can also be permanently altered. And let me just mention something about the allosteric site. So I've already explained what they are, but what kind of molecules are able to bind to them? So molecules that bind to the allosteric site may be either allosteric activators or inhibitors. And the binding of either one will cause a change in the conformation of the protein, but the effects are of course different, because the inhibitors do not let the substrate bind to the enzyme, but the activators make the enzyme more available for the substrate to bind to. We also have covalently modified enzymes. Enzymes can be activated or deactivated by phosphorylation or dephosphorylation. And we don't know if the phosphorylation or the dephosphorylation will activate or deactivate the enzyme, but we can determine that by experiments, of course. Glycosylation, which is a covalent attachment of a sugar moieties, can tag an enzyme for transport within the cell, or it can modify protein activity and selectivity. And the last point that I want to go over for this section is zymogen, and they contain a catalytic domain and a regulatory domain. The regulatory domain must be either removed or altered to expose the active site. So they basically kind of work like a safety pin, and if they are in the right environment, the active site will be exposed so the enzyme can become active. Oh, and also, yes, zymogens have names ending in ogen, and that's how you can identify them if you see a name ending in ogen.